0, 3, 7, and 21. Percentage wound contraction will be calculated using the formula below. Percentage wound contraction, initial wound area minus the wound area on the X times the initial, divided by the initial wound area times 100. Epithelialization time, which is defined as the number of days required to complete epithelial cover of the wound surface without raw areas. Gross wound assessment. Wounds will be evaluated using a semi-quantitative scoring system as shown below. Parameter, you have edema, exudation, infection, and granulation tissue. You have score 0, 1, 2, and 3. Where you have no edema, you score 0, as the table indicates here. Microbial evaluation. Sterile wound swabs will be collected on days 0, 7, and 14. Bacteria isolation and coliform units count. Histopathological evaluation. Punch biopsy samples will be obtained on day 14, fixed in 10% buffered formalin, processed routinely, and stained with hematoxylin and eosine. Sections will be evaluated for fibroblast proliferation, collagen deposition, neovascularization, and inflammatory cell infiltration. Data analysis. Data analysis will be analyzed using SPSS version 20, and descriptive statistics will be expressed as mean plus or minus standard deviation. Differences among treatment groups will be analyzed using one way or another, followed by Turkish post-hoc tests. Repeated measures ANOVA will be used for wound contraction over time. Categorical data will be analyzed using chi-square test. Then statistical significance will be set at the probability of less than or equal to 0.05. Significance of the study. This research has significant implications for veterinary practice, particularly in West Africa, by providing evidence-based insight into the comparative efficacy of these readily available and low-cost topical agents. The study can offer practical alternatives to smallholder goat farming systems where access to conventional veterinary antiseptics and antibiotics may be limited. Improved wound management strategies can reduce morbidity and mortality in livestock, enhancing animal welfare and supporting local economies. The findings could also contribute to the broader understanding of natural wound healing agents and their potential applications even in human medicine, especially in the context of increasing antibiotics resistance. References. This is the application for ethical clearance for the research comments. Thank you very much for listening.
All right. Thank you very much, the presenter, for your beautiful presentation. I think
Thank you,
now
sir.
we go ahead for comments, questions, and answers. So let's give the opportunity to the clean and the seminar coordinator. He's the one coordinating. If people have questions, answers, comments, all those things, so that we listen to them before the chairman. Seminar coordinator.
Okay. Hello. If you have any question, please. You can. If you have any question, please. You can. Hello.
Sorry for that.
Good morning. Good morning, everyone. Good morning, everyone. Can you hear me?
Yes, we can hear you. Go ahead.
Okay. I have some comments to make. That was a beautiful presentation. My first worry is the number of goats. You said 15 goats will be used. I don't know if you are buying them or if you have raised them yourself, but the cost of goats now, they are very high. So I don't know how able you will be to buy 15 goats. My suggestion on that is that I don't know, but I'm looking at a situation where you could create these 3 wounds on one goat and then analyze them. You can have 3 goats and then create 3 wounds on each of them. And then you can have that as groups. Hello?
Yeah, we're
I'm
listening.
listening.
Okay. Then for these 3 wounds on a goat, my take is that that goat is providing the same condition, a stable condition for all the 3 materials you're going to use. So there's not going to be any issue of maybe this goat was sick or this one was like that, but you're going to have a comparison on the same condition of a goat, and then the second one and the third one. First, it will eliminate the number of goats you're going to use. Secondly, it is going to give you a more stable condition and a better result when you compare these ones on the same goat. Thank you.
Thank you very much.
Any other question? If you have a question, you can raise your hand, indicate by raising your hand, so that you'll be called upon to ask your question. I want to recognize the presence of Professor M.D. Sani. He was here with us the last time we had this seminar. He joined the seminar. You are welcome, sir. And we are waiting for your questions for the students. Yes, somebody is raising his hand. And that is Lenvi, Dr. Lenvi Manfe. Okay, you can unmute yourself now and ask your question. Unmute yourself. Ask your question, please. Dr. Levy, are you here? Please ask your question. Okay, Dr. Andrew. Andrew. Please, you can unmute yourself and ask your question. I've asked you to unmute, so unmute yourself. Dr. Raphael Obe, please, you can ask your question. Unmute yourself.
All right.
Yes, please. Dr. Raphael Obe, please, you have the floor. Unmute yourself.
Okay. Yeah. Can you hear me now?
Yes, we can hear you.
Yes, I can hear you.
Okay. Yeah. Thank you. So yes, I could see that you have. Yes, I was speaking earlier, but I don't know whether you heard me. Thank you for your presentation.
Yes.
I saw your objectives, about 6 of them. So can you tell us how you are going to achieve each of the objectives? Just briefly, like the number 1 objective, this is what I'm going to do to achieve this. Then number 2 objective, this is what I'm going to, this is a method I'm going to use to achieve this objective. So please, I would like you to, give us a brief on that, or highlight on that. Thank you very much.
Thank you, sir. The objectives are presented on the slide, and if you go to wound evaluation, for wound contraction, we are going to use a transparent graph sheet. We take the initial wound area shortly after creation. Then at the 0, then the 3, 7, 10, 14, and 21. This the measurement will be compared to the initial wound area, and this will be expressed in percentages. Then that will tell us at each group which group has a very good wound contraction over time. If you go to the second objective, which is let me get to the objective itself, please. The second objective, which is to compare the epithelialization time of wounds treated with these 3 agents. The epithelialization, the growth of this new keratinized cells, which now cover the whole wound after healing. So we are going to take for each group how long it takes the wound to develop the epithelial cells that will cover the wound finally. That is during complete healing. It will measure the number of days that are involved for group A, B, and C. Since they are in group, we take the average of each group to represent the group in number of days. And then the second one is the exudation, to assess the characteristics in terms of exudation, edema, and infection. This one too, a scoring sheet will be provided. When you view it on daily basis, you are able to know which wound has exudate, which one has edema, which one has infection. The one without swelling, you know that there is no edema, and you record it. It also has a score sheet for 0, as it was presented in the other table for wound evaluation. Then histopathological changes. Here we are going to use biopsy samples, which will be analyzed in the laboratory, fixed, and then view under microscope. When you view it under microscope, you will now look at the level of, you now look at the fibroblast that has the fibroblast that have proliferated over time. You look at the collagen deposition and then the new vascular tissues that have formed. Then the macrophages, the inflammatory cells, which is neutrophils, macrophages that have migrated into the wound site. We are going to see them all on the microscope. If I have answered how I will carry out each objective in order to achieve the result.
Okay. Dr. Levy Manfe, please, you can ask your question. Unmute yourself and ask your question. Dr. Levy.
Can you hear me?
Yes, go ahead,
Yes.
please.
Yes, I've unmuted. Can you hear me? All right. My concern is on that microbial evaluation.
Dr. Levy, you are cracking. We can't hear you very clearly. Dr. Levy, we cannot hear you very clearly.
Swabbing the wound at the 07, I think 14, or the surroundings. Because if the wound is not local, okay, send the chat.
Yes, please speak. Okay. Okay, send the chat, please. So, Dr. Andrew, Amine, you have the floor.
Can you hear me?
Yes, go ahead.
Yeah.
Be audible.
It's just a follow-up to what Dr. Patrick said. You're working with key animals, 5 parts, and the entire presentation does not have a budget. It didn't show us. So do you anticipate the cost of this research? You have a lot of things you want to do, and you know they are going to involve money. So how do you, without a budget, how do you intend to carry this out?
Dr. Amine, you will have to take that again. We did not hear you very clearly.
Well, it's
Speak
not
out.
clear. What
Yes.
I said was,
Speak out.
as a part of what Dr. Sabina raised earlier with the number of animals, he did not show a budget for this work. Yet, there are several things he's trying to look at. So my question is, how does he have a grant somewhere? Is he self-sponsored? Does he know how much it's going to cost eventually to carry out this whole process?
Okay. Dr. Cletus, did you hear him?
Yes, I hear him.
Okay, please change the orientation of your phone. Change.
I did not put budget. I did not put budget to this work deliberately because of the changes that happens to our naira every day. You rise up one day, you hear the devaluation of naira, and you are not certain whether the price you have set today will be the same with the one you are going to when you really start the work. So, but I'm believing God that I will be able to, He will sustain me throughout the work. Thank you.
Are you satisfied with that response, Dr. Amine?
Well, when you go, God, now I rest my case now.
Dr. Cletus, that's not the usual thing. That's not the way to go. You must always have a budget. So whatever proposal or grant you are writing, there must always be a budget. So are there any other questions? If not, the chairperson, the prof, you carry on.
Okay.
Yes,
Thank you very much. I hope you can hear me.
Yes, I'm hearing you, sir.
It's all right. Thank you very much for your presentation. I just have a few questions and comments. The first one I want to find out, your is based on wound. You are talking of treatment. So my first question, This wound you are talking about and the treatment, is this strictly applied to only surgical wound or other type of wound? And if so, what is the difference between a surgical wound and any other wound? That is question one. Then your samples, that is the agent you are going to use. You talk about providence and other agents you are going to use. We are very familiar with glucose iodine. So my question is glucose iodine, providence iodine, if no, or yes, what are the differences? If yes, no. The third one is you are talking of 7 and you are comparing honey, aloe vera, providence iodine. So I want to know among these three, which one do you think you serve? Because I learned honey is very expensive, especially pure honey. And that leads to another question. Which type of honey are you going to use? Pure honey, or which one? Then aloe vera, when you compare the cost of getting this, which one will be to use? And also another between honey, aloe vera, and which one is the standard? Or you are just doing a random comparison between the three. Which one among them is standard? And you want to compare the other two? Or you are just comparing each one? Then in your experimental side, you mentioned VTH. I still want to ask because from somebody outside, they say VTH. Those are the ones that are invented. They know it's VTH. But as insider, we have to fight the VTH. The VTH and the VTH South. I think it's important for us to know where the South Pole is and important to. Another point is you are talking of using something about that. And our experience was that most time when people give for experiments, before the experiment come on, a lot of them will disappear. That is, PPR will take them away. Are you factoring that factor in your experiment? The issue of keeping your goat safe against PPR, because PPR has killed a lot of people's research animals. So what are you doing to safeguard against that? Then your experimental procedure, you talk of creating a wound 3 by 3 cm. That is where I don't understand. I'm not a surgeon. So when you are trying to say you create a wound 3 cm by 3 cm, what do you mean? Is it that vertically you cut 3 cm, horizontally you cut 3 cm? Or what do you mean by 3 cm by 3 cm? I think it's very important you clarify that so that people know what you are talking about. The next one is your days of taking samples. You say especially for the microbiological sampling. I think Dr. Levy asked a question about that. You say day 0, day 3, day 7, day 10, 14, and 21. what I observe is there is a 4 and 5 day gap between the days you have given. But my main concern is on that day 0. I want to assume that when you say day 0 is the time before the surgical wound is incised. Then you say you will take sample around the wound area on day 0 and this. So if the 0 is wound infliction, how do you get your sample, your swab around that wound? Because I assume the 0 is before that incision or the wound is created. The moment you created the wound, you are starting as day 1 going forward. So how do you sample your microbiological around the wound site? Is it that you will sample it from the around the area you want to create the wound, or how are you going about it? I think it's important you let us know. My little knowledge of granulation and surgical healing is that epithelialization and other things become active around day 3 and so. But if you see your gap between the 0 and the 3 is 4 days, that is the 0 inclusive. And from the 3 to 7 is 5 days. If the 3 is inclusive. So why the gaps? Why do you choose the 3 and the 7? If you can shed more light on that. And the other one, the issue of cost, I have asked it before. Iodine, honey, aloe vera. Ordinarily, I think iodine should be cheaper among the other ones. How to get the quantity of aloe vera and honey? I think you will need to explain. Then another thing is, you talk of wound healing by second, is it in second intention or what do you say?
Yeah, second intention.
In
Second
livestock, normally we discourage the issues of scar formation because it's a kind of, it defends the animal, it reduces the cost of the skin and hide, though we don't use skin much these days. If it were large animals like the large ruminant like the cow, we can talk about that. But when you create a wound and you allow it healed by second intention or whatever, don't you think it will have a negative effect on the aesthetic value of that animal? Mm. Then the comment Dr. Nu made, considering the number of animals you want to use, she was suggesting that you can create those wounds on the same animal. I was thinking it's something you need to consider, but still considering the fact that creating these 3 wounds on one animal, will it be too much stress on the animal? That is another aspect we have to look at. If it's not too much stress, it will save the cost. That is, it will reduce the number of animals because one animal will be serving as a control because all the 3 wounds are created on one animal. Every environmental and genetic condition is related to that animal, almost the same. So yeah, the result we get from them will be a very good distance. But still, I'm looking at the wafer issue. If you are creating 3 by 3 cm wound, which I'm here to appreciate what you mean by 3 by 3 cm wound, creating these 3 wounds on one animal, will it be too much on them? Because I'm here to appreciate even the dimension of the depth of the wound, the width, the length, and everything. So these are some of the issues I just want you to clarify. And thank you very much.
And thank you, sir. We have lots of questions coming up. And Dr. Cletus, you have to clear this. So related to the last question the chairperson asked, there's a contrary question here. This is from Dr. Emmanuel Mbede. He is saying that while I share the sentiment of cost reduction, by creating 3 different wounds on a single goat, you need to also look at it from the point of ethics and animal welfare, which the chairperson has pointed out. That this concern will arise if you are interested in publishing the findings. So Dr. Cletus, take note. Then Professor Sani has been raising up his hand. Let's take his question. Then Dr. Cletus, you can respond. So Professor Sani, please, let me ask you to unmute. You can unmute yourself and ask your question now. Professor Sani, you can go ahead and unmute yourself and ask your question. Unmute yourself.
You can only send it through message, just like.
Okay.
Thank you very much.
He's up. You're welcome, sir.
Good morning, everyone. Good morning, my dean.
Good morning, Prof.
Yeah, quite a visa.
Yeah, I'm good. Always a pleasure having you. Thank you very much, Prof.
Yes, sir. Actually, yeah, very, very nice, discussion over there and nice presentation. I have few concerns, yeah. As someone made mention, there's what they call there's what they call intra-animal comparative model. Intra-animal comparative model give you room to use a single animal to do comparative study using different treatment. Yes, and there are literature for that. People have done it. And regarding ethical issue, yeah, even it can be published even in international journal. No two way about it. Yes, fine. But then, in doing that, there are a lot of things the student or the candidate have to put into consideration. Yeah, at least the treatment, the treatment in most cases now in his own case now, using more than that wound at 3 different places, and those 3 different places has to be at least 2 centimeters apart. That's what the literature said. At least 2 centimeters apart. The higher the centimeter,
I think the network
in
has,
most comparative inter-animal comparative model, they use acceptable. Yeah, it's acceptable. Yes, to cost. But then, even as there are a lot of literature regarding that. But the problem now is, again, even as at then, you still need to use the minimum acceptable number of animals for statistical evaluation. That one aside. Then, I would like to ask the candidate. The title read, I don't have access to the title now, but it said cutaneous surgical wound. When you say cutaneous surgical wound, you have not said anything. Yes, I know you are in surgery department, yeah, but then at least this is, this is vet and research. You have not said anything. There are different types of wound.
Yeah. Prof, are you still here?
I'm hearing.
Okay. Professor Sani, are you done with your question?
Or cutaneous surgical wound. And in your methodology, can you go to the slide? Is the wound? I hope I've not been talking to myself all this while.
No, I can hear you.
Okay, so I want the student to go to the methodology where he created the wound, because he need to tell us the type of wound. Yes, he created. Just go. Let me see the slide.
Move your slides to the methodology. Yeah.
Okay. You said under accepted condition, is standardized full thickness skin wound measuring this created on the. You have not said anything. You are in surgery. You should know. So that
Yes.
wound now, and you are saying 3 by 3, as I think they didn't even mention. You see, you have to qualify the type of wound. A cutaneous surgical wound could either be incisional or excisional, and it can also be combined, both incisional and excisional. You have to tell us so that we appreciate. You cannot get a 3 by 3 with an incisional wound. You can't get it. You can only get 3 by 3 when you do an excisional wound. You remove part of the skin. Are you removing any part of the skin? Just answer a quick one. Just answer, please.
Yes, we are removing.
Yes, sir.
Yes, sir, we are removing.
So to we name an open wound. Hello, are we together?
Yeah.
Dr. Cletus, please begin to respond to the questions raised by the chairperson.
Thank you
And
very
that's all.
much, ma'am.
Please
Yes,
return your.
I will start with the differences between surgical wounds. There are other wounds, as we all know, but surgical wounds are intentional injuries that we create during operative procedures on the animal, and they need a very good care or management for them to heal. So they are different from other wounds that may be caused by maybe accident or other trauma or agents that can create them. So they are quite different. They have smooth edges. They can be managed easily, more than when other wounds are involved. You talk of Logos iodine and povidone iodine. Yes, povidone iodine is quite different. It's a preparation that has iodine complex with polyvinyl prolidone. This complex, when it comes in contact with wound exudate, at that point, iodine is now released into the wound. So it's quite different from Logos and other iodine formula that are in the market. So povidone iodine, if well prepared, is safer to use on wound than these Logos or other iodine that are there. Considering the price, which of these three agents that are cheaper? The essence here is not only looking at the price of these agents that we are working on. We are also trying to identify which one is safe, the safest and effective that we can use on these breeds of animals that obtain the best result. So even though iodine is very cheap compared to the three agents, it has some cytotoxic effect on fibroblasts and epithelialization, thereby causing delay in wound healing, which could lead to several costs and even death. So that is that for that. You said in Uniagric we have VTH short core and VTH annex. This experiment is going to be conducted at VTH at short core, not in annex. The wound 3 by 3 cm dimension that I have mentioned in my procedures, it means that the wound will be a 3 by 3 square. That is what I mean by 3 by 3 cm. I will remove part of the skin and leave this wound open. I know due to pressure and contraction, the wound is not going to remain like a square wound. It will still, the wound edges will still constrict and maybe expand more than this 3 by 3 cm by sight. So that is what we intend to do. Then taking samples or measuring the wound at the zero. The zero here, I mean, that very instant the wound is created. To me, that is what I refer to as the zero. Then I will take the side of the dimensions of the wound, and then on the zero. And this the zero will serve as a reference point in order to calculate wound contraction, which will be expressed in percentages, and then be compared over time with other groups. That is what I mean. Wounds. There are several wound can be healed by first, second, and third intention. In surgical wounds, when you stitch them, the wound edges are brought together. There is a position, and this healing occurs faster, and there is there is less or no scar formation. But when wounds are healed by second intention, definitely there are scar formation that will be left after healing. The scar formation, based on other research conducted, is also affected by the wound healing agents that you use, apart from the size of the wound that is involved, or the tissue that the amount of tissue that are involved. So looking at these 3 agents in terms of second intention, we are still trying to get which one that will give us the best result. Best result here means minimum scar formation and then quick or early healing that will also not disfigure the animal concerned.
Okay, Dr. Cletus, there are some microbiological concerns here. One of the questions is, very quickly, you answer very quickly. Because we have
before you go forward now, coordinator, please.
Yes.
Let's clear this issue. As it has mentioned, if you're day one, when you are talking about this is a standard experimental procedure, and we need to be clear about it. Before somebody will listen to the University of Agriculture, just as I want University, Macaulay, they say day zero is the day you created a wound. In every parameter you are going to measure,
yes,
the zero is pre that event.
Okay.
Whatever you do before you start is what is referred to zero. In fact, in most cases, if you are using those animals as a reference, you take your zero as your reference before you induce anything you want to induce. If you are talking of the zero can only apply here if the site at which you are going to make your incision, you just want to swap around site. But at that time, you have not created a wound. By the time you create a wound, that is your day one. So take note of it can be day zero.
That's all right, sir.
Okay. All right, thank you, sir. So the question is, if you are swabbing the wound, will that not affect the wound healing process? Someone is asking from microbiology. If you are swabbing the wound, you mentioned you're going to swab the wound. Will that not affect the wound healing process? And the second question is, what bacteria exactly are you considering as contaminants, considering we have normal skin flora? I hope you got that. Then an adjunct to that is, what extent of their population will you consider as contamination? Know that they are normal skin flora, and what would you consider as contamination at this wound site? Please respond.
It's all right. The swabbing of the wound will not cause or interfere with wound healing, in the sense that before you swab, I will use distilled water, dip my swab into it, and it will be soft. And then a gentle roll over on the wound will now pick the bacteria or whatever contaminant that is there, which will not create any trauma to debride or create any trauma that can lead to delayed healing. The infection, the colony of the bacteria or whatever the bacteria that we are going to consider effective, or I mean, we are going to consider here will depend on the number of colonies that are seen on the culture media. So if there's a colony, we consider that the wound is not is mild. We have moderate, and we say I have severe infection. So for colonies that are just scanty on the slide, you take it that there's mild infection, there's severe infection, and there's moderate infection. So in terms of the bacteria or the microorganisms we are looking at, the research work does not particularly look at the type of microorganisms that are involved in this contamination, even though one of my objective is to isolate them and then be able to identify, consider the infection as being mild, severe, and moderate. So we are not targeting a particular microorganisms to consider the wound being infected. Thank you very much.
Is there a reference standard for your consideration for mild, moderate, and severe infection? Do you have a standard reference that you are, comparing what you have to? Because this is my first time to hear about this. Is there a standard for that? For you to say, you said something about when you look at it, when they are very few, you say they are scanty. Is that your own metrics, or there is a standard for that?
There is a standard, but it has gone off my memory now.
You have to consult with microbiology, microbiology expert in that area, so that you do is acceptable. Then one very quickly one, will you assess or evaluate the sterility of the treatment agent? That's the honey that you're going to use. How will you assess that the honey you are using is sterile, that you are not introducing contamination by yourself? Very quickly, please.
Is it right? Honey is has antimicrobial activity, which are very, very high. There is no way any microorganism will survive inside honey because of the high level of sugar that is there, which drains water from the microorganisms, from their cells, and then causes dehydration. So even if they are already none of them will be alive by the time you are applying it. So that rules out bacterial contamination from honey entirely. There are other areas that are.
Go ahead, please. Go ahead.
There are other activities, antimicrobial, anti-inflammatory, but that one is until when you apply it to the wound. So, but in terms of contamination, that is that.
Okay. Professor Victor Ahu wants to ask a question, so we'll take that will be the last question we're going to take. Prof, please unmute yourself and ask your question. You can unmute yourself and ask your question.
Hello?
Dr. Victor, yeah, okay, we can hear you now.
Okay.
Yes.
Okay, all right. Okay. My contribution is on the reference to the 0 of the start of the experiment as raised by the dean. To me, I was looking at it that this is an experiment that is comparing the effects of wound. You have to create a wound, and this is related to the treatment. So if you create a wound and then you've not started treatment, that could easily serve as your day 0, because it's the treatment that matters in this form of experiment. So if you're considering just the skin without a wound as to be your day 0, I think that could be a different experiment in such a measurement, in such a parameter that is going to be taken. So that's my concern on that discussion. Because when it was said, the presenter accepted it, and I was a bit worried that maybe probably there could be some more discussion on that, because that matters on the points parameters are going to be taken. So we need some clarification from there concerning the 0 that the presenter and the dean were making reference to. Thank you.
So very quickly, Dr.
Make a comment before we go ahead.
Okay, sir. That is just related to a response he gave. The statement he made that honey cannot be contaminated by bacteria is not correct. Is wrong. We have Dr. Emmanuel Mwede here. He has put up that response here that is not correct. We know very well that Clostridial spores can be found in honey. So it's not correct to say that all honey cannot be contaminated. Please, Dr. Cletus, take note of that. Chairperson, sir, over to you.
It's okay. It's not all bacteria that can be infected by honey. The point I was making is, even without applying anything, the tendency is that the body has the ability to heal itself. All you are doing is you are helping the body to heal. So the instance you create that wound, healing have commenced. Because injury you have everything has gone. So that cannot be your day 0. It is only your day 1. It can't be day 0. You have not started treatment. So just taking that as the 0 may not be what we have done. The 0 is 0. It may have some value at a certain time, but it's like a starting point. When you start from 0, you go to 1, and then you continue. There is another question somebody was asking. If you have located the site you want to do your surgery, you size your wound, and you have taken a swab of those areas. By the time somebody was asking, how do you know these things are normal flora? If you have taken a swab of those areas, all those microorganisms you have found around that area, you can assume the normal flora. By the time you add swap and you still find them there, you can compare them with what was there before even the surgery. That helps you to know that nothing new has come. So the point I'm making and the point I want to inform and the point still remain that day zero should be pre-event. Not after you inciting your wound and you are saying it's day zero. It's the day you start treating that you say is day one. The body is responding even if you are not treating.
Yes.
Thank you.
Right, sir.
Information, clarification. Hello.
Yes,
Yeah.
we're with you.
Dr. Cletus, quickly, please.
I am with you.
You said you wanted to give us information.
No, I'm not the one that have the information.
My network today is not comparative. Bad
I'm
network over here.
not the one with the information.
I think it's,
Okay.
it's Professor Sani that is saying he wants to give information.
Yes.
Okay.
Yeah.
You see, actually, I've listened to that with regard to that the 000. So actually, that if I got it very right, that zero, that means the problem with the zero value is that, yes, that is the diameter of the wound. That is after creating the wound. It's necessary you take the diameter of the wound after creating the wound. And that diameter of the wound, which might be, let's say, 5, the diameter maybe is 5 mm or what have you, of the circumference of the wound. And that wound now, that diameter now at that 0 will serve as the control. But the implication now is that at day 1, he can only compare it with that day 0. Then day 0 will now serve as the control for the rate of contraction of the wound.
Exactly. That was what I was also thinking of.
So that is the only thing. Yes. So that day 0 now will serve as your control, just like you appear person for the treatment and also control. Then at day 5 will be compared to day 0. At day this will be compared to day 0. But then when you are doing that, that means for every comparison, that day 0 value will still remain with that same diameter as control.
Yes.
That is what is as in wound healing when you are using day 0. Yes. So
that was my line of thinking.
Then, as Dr. Nancy really said, give us, because I'm really interested in knowing the reference you use for the scoring. And yes, because I'm really interested to know that reference you use for the scoring. And how do you intend to show? Okay, this is this one is infected, this one is mild. Or that reference at least is very good. And apart from in wound healing, you see, cutaneous wound healing in goats depend on so many complex network of biological markers. And it has been suggested that, yes, for adequate wound to heal, there should be the level of serum protein should be high. When you have low level of protein, that means there is catabolic process going on. And with that low level of protein, that means it will definitely delay wound healing and the wound strength, including the strength of the wound. So if just one marker, protein or albumin, which is very, very essential, and that protein the level serve either as catabolic or anabolic process. If you can measure that one alone, at least, that will add more to what you are doing. Then again, he made mention about povidone iodine, and I'm not satisfied how he explained povidone iodine. Povidone is not a drug. Povidone is never a drug. Povidone is a carrier. And iodine is incorporated into the carrier, which is the povidone. So, and what it does is that when when you want prolonged, sustained release of the iodine will be released slowly into the tissue for a long time. Instead of just applying the iodine, you are now incorporating it into a carrier, which is the povidone, so that it can sustain it for a long time. So that is what we do. And you can get it. You don't have to even prepare it. I had you say preparing. There are formulation of it in my office. I have it myself here in Zaria. You can have it. It can be in ointment, can be in cream, povidone iodine. So, and we did a research on the same povidone iodine. People use it to treat some infection, bed infection, Newcastle, Outerview. We have it. So that is just my one or two comment, and other ones, maybe if the student can reach up to me, at least I can tell him more with regard to wound healing. Is something we have done several times, and we have compared several things. And as you are saying, this comparison he's saying, I can give you 5 literature that have done comparison with honey and povidone iodine. Not that it has not been done. There are several studies that have done the comparison. So we don't have to go there now, but there are several, I can give you several literature with that. And we know the outcome of those results, what they have done. So you repeating it is not a bad one, but you have to employ certain ways of assessing the wound healing. What those previous work has not done. And that's why assessing 1 or 2 markers, that enhance inflammation, that enhance repair, or decreases inflammatory response.
Pastor Victor, please, you can go ahead and make your last statement to this discussion. Thank you.
All right, good. Thank you so much, Al. I was listening to what MD Sani was saying concerning that's the biochemical parameters that are also for factors that are also needed in wound healing. So to me, I was thinking before these animals are going to be used, they are going to be ascertained whether they are apparently
There
healthy.
cannot be wood
So some of these parameters. Hello. So to me, I was thinking some basic biochemical hematological parameters will be taken even before the animals will be used to ascertain that they are apparently healthy. So if all the biochemicals or the biochemicals that are needed, that's albumin, fibrinogen, and all these ones, if they are there and they are needed for wound healing, the basic parameters before these animals are going to be used should be ascertained. So those factors can already be taken care of even before the creation of the wounds. So thank you.
Okay. Thank you very much, sir. Chairperson, I think we'll allow the supervisors to now.
Thank you very much for the comments people have given. Just like the last speaker said, if you are talking of taking those parameters before the wound is created, when you took these parameters before the wound is created, what do you call them? They are parameters taken at day 0. So the issue of day 0, we still have to look at it critically, because if your intention is to compare these things with the time you started, you are using the initial time you created the wound as your day 0, and you want to compare everything on the basis of that, yes, you can go ahead. But in the real standard experimental setting, when you talk of your day 0 always refer to pre-exposure of the animal to this thing. Like in pharmacology, if I want to induce diabetes in an animal, I take the parameter on the 0. That's it, before I induce anything. Because once you have created a wound, you have induced something into the body, and a lot of changes will come as a result of that. So when you say it refer to your 0, you really, really need to clarify what you mean by the 0. If not, that confusion will always remain. So it's better we take care of that and know how we move forward. I think we want to appreciate everybody for the contribution. Professor Sani from Zaria, and all the people that have contributed. This is one of the things you want to achieve. Since it is a proposal, we want people to argue, make contributions, so that at the end of the day, the work will be much, much better. It's a filtering process, and I think we have contributed. Now we will give room for the supervisors. If they have some few comments to make, then we move ahead. If you agree, the coordinator.
Yes, sir, please. The professor and the supervisors are supposed to say something. Thank you, sir. Professor Esther?
Yes. Thank you so much for your contributions. the zero of physiological values, they are the same.
Oh. Okay.
Yeah.
Okay, take parameters. After creating the physical rule, as was mentioned by others, we are going to take measurement. And that measurement is going to be served as the control, comparing with other tests we have from later. And getting 3 by 3 cm wounds on the body of the animal, that means you are creating a square type wound on the body, square type of wound we are creating. That's it. So considering that we are going that let's use one animal and create 3 wounds on the body. No, that's no, please. Because we know that once you apply a topical drug on the wound, it's going to absorb in the body. So the 3 are going to also at the same time, it's going to cause problem. You won't know which one is effective, which is not effective. That's why we don't buy that one. We use animals, one animal per wound. So 15 animals, we are going to buy 15 animals after we sell the animal out. So we go back our money. So thank you so much for your contributions from all of us. We will be with you.
Thank you very much. So, coordinator, over to you.
Okay. I want to appreciate every contribution so far towards this seminar, towards this presentation. Congratulations to the presenter and to all of us. So the second presentation is an exit seminar to be presented by Dr. Peter Ochala. Dr. Peter, and to chair this seminar is Dr. S.N. Nom Adam. The student has 25 minutes for his presentation. Dr. Peter, please share your slide. Ma, I hand over the proceedings to you. Thank you. Dr. Peter Ochala, share your slides, please.
Thank you, coordinator. I want to acknowledge the Dean, College of Veterinary Medicine, Justin, and I
Thank
also
you,
acknowledge all the professors present at this presentation. Regards to the postgraduate students, we have an exit seminar to be presented by Dr. Ochola Peter in the Department of Anatomy. The title is Histomorphological and Immunohistochemical Studies on the Female Reproductive Organs of Juvenile African Greater Cane Rat. So over to you, Dr. Ochola. You have 25 minutes.
Good day, Dean College of Veterinary Medicine, the directors, professors, HOD Veterinary Anatomy, the seminar coordinator, lecturers, staff, fellow students, amiable online viewers. I'm Dr. Peter Ochola Friday, and I'm here today to present an exit seminar titled Histomorphological and Immunohistochemical Studies on the Female Reproductive Organs of the Juvenile African Greater Cane Rat. The African Greater Cane Rat is scientifically known as Theonomys vendoranus. It's an important one rooted in the sub-Saharan Africa. It is valued for its ecological role, economic importance, and as a protein source. And the animal has a growing potential as a laboratory model, recognized in different languages, in TIP languages as Bia, Idoma, Obije, Jukun call it Aje, Yoruba call it Oya, and Igbo call it Inche, and Hausa call it Kuntutu. All are in Nigerian vernacular languages. This is a picture of the the African greater K rat. The statement of research problem. Histological data about the African greater K rat, juvenile female reproductive organs such as ovary, oviduct, and uterus and vagina are not yet documented. Data on key cellular inflammatory markers important in reproductive organ development, somatostasis, angiogenesis, such as calcium and CG125, interleukin 6, interleukin 8, are lacking. There's a lack of immunohistochemical profile for S100 in the juvenile female African greater K rat reproductive organs. Justification for the study. Even with the relevant studies, there is a notable lack of information on the reproductive system of juvenile female African greater K rat developmental stages that can influence adult fertility and reproductive success. Histological and immunohistochemical profile will provide reference standard for veterinary practitioners working with African greater K rat. Therefore, this study is justified as to contribute novel information to the field of African greater K rat reproductive histology, supporting the sustainable development of the African greater K rat farming. Aim of the study is to carry out histology and immunohistochemistry studies of the female reproductive organs of the juvenile African greater K rat. Our research, the research objective is to study the normal histology of the parts of the female reproductive system, including ovary, oviduct, vagina, and uterus, in apparently healthy state using hematoxylin and eosin staining protocol. Also to study the mucin distribution by employing Asian blue, which is for acidic mucin, and periodic acid chip staining for neutral mucin techniques. To study part of the female reproductive organs immunohistochemical using 4 major biomarkers, which are S100 proteins, calcium CG125, interleukin 6, and interleukin 8 biomarkers. The top questions: what are the histological features of the ovary, uterus, and the vagina of the juvenile female African greater K rat? How are neutral mucopolysaccharide, glycogen, and acidic mucin distributed in the juvenile African K rat reproductive organs? What is the pattern of expression of S100 protein, which is a neural marker, calcium CG125, interleukin 6, and interleukin 8 in the reproductive organs of the juvenile female African greater K rat? Significance of the study. The study will provide first detailed histological baseline data on the reproductive organs of the juvenile female African greater K rats. It will advance knowledge on the neural, epithelial, and immune regulation of the reproductive tract before puberty. This work positions the greater K rat as potential comparative model for studying juvenile reproductive development, mucosal immunity, and gynecological conditions. Materials and methods. Animal procurement and management. 6 apparently healthy juvenile female of 3 months old were procured from Golden Boy Farm, Apodus, Abuja, FCT, Nigeria, and transferred to the research laboratory in the Department of Veterinary Anatomy, Joseph Simon Taka University. The ethical contribution and approval was sought and obtained from the Animal Ethics and Welfare University with the ethics number Johnson CVM Ethics 2025-09. Here is an attached copy of the approved ethical clearance. Materials and methods, tissue collection. After the animal were anesthetized and stabilized, they were anesthetized using the carbon dioxide protocol by the American Veterinary Medical Association 2001 guidelines for rodents, euthanasia rodents. And there was a surgical incision through the linear alba to the abdominopelvic region to expose the reproductive organs. And these tissues were carefully excised and they were preserved in 10% neutral buffer formalin for histology and immunohistochemical study. The routine process of the paraffin embedded embedded paraffin tissues, 5 microgram serial section, staying with hematoxylin and eosin for general application according to Gambo et al. 2008 method. Ferrosic acid shield was also staining was carried out to detect neutral mucopolysaccharides in glandular epithelium according to Bancroft and Lee 2019 method. Asian blue staining was also done at a pH of 2.5 to detect acidic mucopolysaccharide. The result is here. This is plate 1. It's a photomicrograph of ovary as times 100, and what is observed is presence of primordial follicles at this age, secondary follicles, zona musculosa, and we have the oocyte, blue arrow, Graafian follicles. And we couldn't find corpus luteum, which is an indication the animal has not yet, there's no population yet. The second slide was that of a section of the infundibulum. At maturation of X100, the stain used was X, H, and E, and the indications, there's a penetrator oviduct, which is the infundibulum, granular cell, follicles as yellow arrow, and then secretory duct blue arrows. Another section of that is that of the oviduct, which is prominent lumen, blue arrow, and then musculary mucosa with the black arrows indicated, secretory glands, yellow arrows, H and E as 100. And then we proceeded. Also, we did that for the pictures too. Endometrial gland were indicated with the black arrow, and the dense connective tissue was seen. Musculary and blood vessels were also observed in these studies. And the vaginal section here will be hypersecreting and high plastic glandular epithelial cells, which is a single squamous epithelial, and muscularly connective tissue or stroma were also observed in the study. The next study was that of the special stains. We conducted special stain studies using PAS, periodic acid chip, and Asian blue stain. And the table, some of the slides are here. Let me go straight to the table. The table here is a summary of the stain using periodic acid sheets. The ovary indicate positive, which is an indication, a neutral, meaning there's a neutral mucopolysaccharide secretion. The oviduct was also assigned positive, mild mucin secretion, uterus high secretion of mucins, and then there's a mild secretion of neutral mucopolysaccharide at the vagina. Then that of the Asian blue, the granulosa cells and zona pellucida oocyte cytoplasm and the surface epithelium was highly positive, which is a sign of high secretion of acidic mucin. And then that of the oviduct, acidic mucin was secreted, but it was mild. Uterus has a mild secretion of acidic mucin, and vaginal had a high secretion of acidic mucin. Immunohistochemistry was also conducted, and the photomicrograph here, the plate is showing the diffuse expression of epithelial cells, strong expression of neural cells, and very weak expression of stromal cells to S100, which is a neural marker. And then that of the second slide is reading that of calcium at 125, which shows ovary being positive stain for epithelial cells. The table here summarizes the immunolocation of the biomarkers. The calcium antigen 125 was localized at the epithelial cell of the ovary. The interleukin 6 was localized at the follicular cells and that of the stromal cells. Interleukin 8 was localized at the stromal cell. S100 was localized at stromal cell and the nerve fibers. That of the juvenile, the oviduct also at the stromal cell, calcium antigen 125 was localized at the stromal cell. Interleukin 6 at epithelial cells and the lamina propria for utopia. The interleukin 8 was localized at epithelial cells, lamina propria, and the S100 was localized at lamina propria, musculari, mucosa, and the nerve fibers. The same thing is applicable to that of the uterus, and it's as indicated in the slide. You can see the Acacia 125 was localized at lamina epithelium, and that of the stroma cell. Interleukin 8 was at lamina epithelium, glandular epithelium, and then stroma. Then S100 was only localized at myometrium. For the vagina, the calcium 125 localization was at epithelial linings, which is a simple squamous epithelium, and the calcium the interleukin 6 was at stroma cell. Interleukin 8 they took place at epithelial cells, stroma at the stroma, and then the S100 was localized at stroma and the nerve fibers. At this stage, discussion. At this stage, the primary germ cells and secondary follicles are present, indicative of folliculogenesis. And this was also reported by Cora et al. 2015, reporting similar ovarian artificial in mice and laboratory animals. There's a moderate positivity observed in this study suggests neural mucin presence, which agree with the finding of Wandinga et al. 1988. There is a strong Asian blue positivity means abundant acidic mucin, which contribute extracellular matrix formation, and as reported by Roger and Interweb Roger in 2015. Cells were noticed to be prominently reactive to S100 but less reactive to interleukin 6, interleukin 8, with the minicellular growth. Cuboidal epithelial cells were moderately reactive to calcium 125 marker, indicating of cellular protein presence for ovarian physiology. The simple ciliated columnar epithelium was observed at the level of the oviduct, supported by connective tissue stroma, well-developed muscularis mucosa. The presence of ciliated epithelial cells suggests that the oviduct is structurally prepared at this stage for future gamete transport and fertilization. Strong Asian blue positivity indicates abundant acidic mucin within the oviductal secretion. According to Jensen 1995, acidic mucin play important role in lubrication, protection against pathogens, and regulation of microenvironment required for fertilization. Interleukin 6 and 8, immunoreactivity observed in both the epithelium and lamina propria suggests active cytokine production within the developing oviduct. Similar observations were reported by Rosin et al. 1996, who demonstrated cytokine expression in oviductal tissues involved in epithelial regulation and local immune defense. For the oviduct, S100 expression in nerve fibers, and there's a weak expression within the lamina propria and muscularis, indicating the presence of neutral regulatory mechanism within the oviduct. Similar findings have been reported by rodents, where S100 positivity neutral elements contribute to smooth muscle coordination and security activity, as reported by Kone et al. 1990. The uterus consists of a simple columnar epithelium, endometrial gland, dense connective tissues, stroma, blood vessels, and a well-developed myometrium. This observation agree with the description of juvenile uterine architecture reported by rats and mice by Sato et al. And Gravis 2012. The vagina consisted of glandular cell, connective tissue stroma, and muscular layers. The overall morphology corresponds with the description of juvenile rodent vagina reported by Cora et al. In 2015. Mild pars positivity was observed, and the secretion indicates the presence of relatively small quantity of neutral mucin and glycoprotein. Anderson et al. 1984 reported that glycogen accumulation increases skinary following puberty and estrogen stimulation. Strong Asian blue possibility indicate abundant acidic mucin within the vaginal mucosa cell. Acidic mucin are known to contribute to lubrication, moisture retention, and antimicrobial defense according to Ogara and Faden, 1988. Calcium 125 possibly observed within the vaginal epithelium suggests active epithelial differentiation and maintenance. Similar findings have been reported in malaria-derived reproductive epithelia by Jacob and Bass, 1989. For interleukin 6, the expression is within the stromal cells, which indicates active immune regulation and tissue development. According to Wera et al., they reported that cytokines within the vaginal stroma contribute significantly to the mucosal immunity. For interleukin 8, the expression was observed by both epithelial and stromal cells, which suggests active chemokine-mediated immune defense mechanism. Similar findings have been reported in reproductive mucosal tissues, where interleukin 8 participate in lymphocyte recruitment and epithelial protection, according to Jabo et al. 2009. Weak S1 expression within the stromal cell and strong expression within the nerve fibers indicate that neural elements are more immunoreactive than connective tissues components, reported by Danoto et al. In 2013, who described S100 as a marker of neural tissues cell involved in calcium mediating signal. Immunohistochemical analysis revealed that interleukin in 6, interleukin 8 are juvenile reproductive tissues, indicating active cellular process, and this agrees with what FC Cloudgate 2016 reported. The localization of this marker suggests spatial regulated protein expression associated with tissue differentiation. And this is in agreement with what Vera and Miller 2014 reported. This finding demonstrated that molecular signaling pathways are active during early reproductive organ development and not until when reproduction starts. Comparison with previous findings, Yoba and Nadamun 2018 describe histology and organization of the female reproductive organ in adult African greater Kenya, and they reported a well-developed epithelial lining, stroma component, and glandular structures. The similarity with this current research is that there's an organized tissue architecture observed in this study. There's the presence of these 6 epithelial layers and connective tissues, but there's a clear structural organization of the reproductive organs was observed in the adult, while this place was observed in the juvenile state. Comparison with Aldo et al. Okele 1987, previous finding, he reported that ovarian follicle at various developmental stages, and they also identified well defined ovarian stromal tissues in grass cutters. Similar finding was found in this current research, where we find developed follicles, organized stromal components, and it was also identified. This study demonstrated these structural features during juvenile life instead of that of in the adult life that was reported by Ekole 1987. This highlighted early establishment of reproductive tissues organization. The results of this study have important implications for understanding reproductive development in African greater Kenya, establishing baseline immunohistochemical profile for S, interleukin 6, interleukin 8, calcium 125, and S100 in juvenile reproductive organs is essential for identifying deviation associated with disease and environmental exposure. And this was also reported by Cook and Walker 2018. The cytokine expression pattern observed in this study provide insight into the inflammatory immunity regulation. Recommendation: they should conduct comparative study across developmental stages, apply molecular techniques such as gene expression, investigate additional markers to harmonize regulation, profile, and differentiation of deeper functional insights. Contribution to knowledge. This study is the first comprehensive investigation to combine histology, histochemical, and immunohistochemical techniques to characterize the female reproductive tract of juvenile African greater king rats. It provides novel baseline data on tissue architecture, mucin distribution, and localization of calcium 125, S100, and interleukin 6 and 8, thereby expanding current knowledge of the reproductive development and physiology in this economically important rodent species. Limitation. The study will have benefited more from hormonal assay, complemented by finding of the study. The study was done ex vivo, and an in vivo study will afford a real-time assessment. There's also a cost limitation. Here are some of the references attached here. Thank you for listening.
Thank you, Dr. Peter Ochola, for a wonderful presentation. I would have been more comfortable if this study was a little bit comparative, but it is good you have given us a baseline data for study on reproduction. I personally would have loved to know what it looks like towards estrus, towards puberty. The age of puberty is 6 months. You bought 3 months. I would have loved to see what will be happening in 4 months old and 5 months old and the 6 months old. Then we would have had a very good representation of these techniques you are talking about. That is my first comment. My second comment is that, well, this is an informative research. I think this research is important to researchers. Future application applies to researchers, unlike what we have in cytology, where we can actually see the cells at each stage and identify. Because if you are in reproduction or you want to produce these animals in bulk, you want to know the estrus, the signs towards estrus. But of course, this is a different research, just giving us information on these techniques. Thank you very much for your contribution to knowledge. I will throw the floor open now to questions. Hello. So please, if you have questions, you can indicate by raising your hand and then follow. So Dr. Victor is asking to, he's giving you time to ask a question. Please unmute yourself and ask a question. You can unmute yourself.
Hello?
Now.
Yeah. Okay, am I audible now?
Yes.
Okay, let the presenter take us back to the first slide. That's the title slide. All right, so good. The title reads Histomorphological and Immunohistochemical Studies. Well, I just want to ask in simple terms, what if it is just clearly saying histological studies? Because to me, well, in my very little knowledge about immunohistochemistry, I think it's just a staining process, a staining technique. And that is also under histological studies. So why the complex in the title of the study? Is it to make it look very big, just on my side of this thing expression. That's my first question. I'm still doing something. I have other ones, but I'll raise my hand after some time. Thank you.
Can I respond now? Go ahead. Thank you for the question. The study cut across histology, which is the normal tissue architecture. And that's what the aim of doing the normal histology is, to just reveal the normal tissue architecture. And without histology, you cannot proceed to immunohistology. Histochemistry is also
okay, sorry, my worry. Sorry, guys. My worry is histology is a field of study. Is that okay?
I'm with you, sir.
Okay, histology is also a field of study. You're studying cells at the cellular level, and before you do that, you stain them in order to have some expressions, to see some certain features. Because without staining, you might not be able to identify the different cells that are in the field or exactly. So to me, immunohistochemistry is a staining procedure, it's a staining technique. So, but given that it's a title, because you're studying the histology of juvenile, the reproductive tract of the juvenile African giant cane rat. Is that okay? So, but by putting the staining technique as if it's becoming a title, I think you're doing a histological study of those reproductive organs in juvenile and cane rat, isn't it?
The study is not just limited to the normal histology of the organs. We intend to bring out, we did a immunohistochemistry, which brought out a carbohydrate expression in each of the organs. And that process where immunohistochemistry comes in, histochemistry comes in, rather. The immunohistochemistry is now the method that involve the use of the biomarkers, like the interleukin 6, interleukin 8, and calcium anti-125 and S100. That's where they come in this research. So you have like 3-in-1 research now. You have normal histology. You can choose to stop at the level of normal histology. You now extend to protein carbohydrate expression, which is the histochemistry now. And when you now go further to molecular level, bringing protein expression in, that is where the immunohistochemistry comes in these studies. So each of them can stand alone as a research. The normal histology, the histochemistry that determine the carbohydrate expression, and the immunohistochemistry can also stand on this one.
All right. Well, I'll talk to your supervisor more on that. I'll have a discussion with the supervisor. Thank you.
Dr. Baka, I can ask a question.
Hello? Hello?
Can hear you, sir.
Okay. It's all right. Thank you very much. Thank the presenter for a very beautiful presentation. Just mine is supposed to be a comment, more or less. But as the sentiment of the first person that asked the question, histomorphological immunohistochemistry. And I think the title should have looked at what it did. More of histology, the histomorphological that is there, that I don't quite understand, that I wanted more light should be shed on that. And the other aspect I want to comment, I didn't listen to them very well when they were presenting because it was on and off. But the comment I want to make generally is there is a trend I'm observing, and I think it's not good for our college because I know somebody in this college and in that department of anatomy worked on this greater Kenya, so to speak. Professor Bada,
Yeah.
he did morphological and other things in the reproductive and even the brain and all these things. I remember in those days, myself, him, and Dr. Noja used to travel to Otukpo to go and get these animals from that David Mark farm in Otukpo. Based on what he did, he was even invited by NVMA to present a paper on micro livestock in the continuing education of 2010 in Mina. We were there. And in all the references the presenter put, I didn't see where he referenced his work. And I begin to wonder, are we promoting ourselves or are we demarcating ourselves? Where a colleague in a particular department has done some work in an area that you are working on, you people behave as if nothing has been done before related to that area. I don't think it's good enough. I don't think it's good enough. So I think the supervisory team I learned is the HOD, Dr. Raiche, and Professor Sam. But there is so many works on that. Some of them are published. I know we publish together in that department. And for them working on that the same animal and not acknowledging that work, I don't think is the right way to go. That's just my general comment. You can't be looking for references of all. I know the Yeboah references he mentioned here, but they will quote that person as well. And if we have local materials that are suitable and available and are good enough, we shouldn't be going to be looking about some of this. I just want to make this comment. I don't think that is the right way to go. That's just a general comment I want to make about this. There are a lot of publications from that department that are available online. And for them not to make reference to it and behaving as if it's something entirely new. And I know most of them, they must have read that book. I think it's not the right way to go. That's just my comment. Thank you very much.
Thank you, sir. Dr. Rapalobe, please, you have the floor.
All right. Thank you, coordinator, and good afternoon, everyone. Thank you, presenter. Can you hear me, please?
Yes, sir.
Okay, all right. Thank you, presenter, for a wonderful presentation. I will start from what the Dean just said, because I have a question earlier, before you concluded that what is your contribution to knowledge in the field of study, in this field of study you worked on? And I saw that you gave your contribution to knowledge, and you mentioned the fact that this is a baseline, values you were able to obtain. So now, if a senior colleague, in the college, Professor Badaya did work on this greater king rat, because I remembered, when he did that work, I remember this very animal, but I was wondering whether this was a new animal. So now the question is that, does it mean he doesn't have any baseline value? I mean, when he did his work, he did not get any value related to, what you just presented, or is it that the work he did is completely different, from this one that you did? Because I thought that he also worked on reproductive aspect, reproductive, structures or organs of this animal. But when you now say that your work is just informative study, providing baseline values on reproductive organs and other, aspect, structure of the animal. So I begin to wonder. So the question, take note of this question now. Does it mean that from the work he did you come across his publications? And is it not related to these reproductive organs that you just studied and provided baseline values? That is one. Then the next would be I saw uterus. Uterus, with O, with O starting, alphabet, or the first letter is O. Another one, uterus with first letter U. So I started wondering, is there a difference between these two? Because the pronunciation is similar, uterus. So, but I know of uterus with U as first letter. So, but the other one with O as first letter, is it a different term? So if there's a difference, please, I would like to know the difference between the two. Then, this issue of your title. You see, I wanted to understand something, Dr. Victor Ahu was trying to, make a point, give you information, but your explanation really did not, contradict what he said, because all that you have done is still histological. There's histology in it, because even the immunohistochemical study you mentioned is still histology. So you see, when you form title, when you are framing title, if all that you are doing is under histology as a broad topic, a broad field of study, then it's still histological. So you are going to have different methods and techniques and different aspects of histological study. But it doesn't change the fact that it's still histological, because even from the title, there's histology in this, two aspects you try to separate: histomorphological and then immunohistochemical studies. So it's all still under histological. All right, so please take note. Maybe you have to look at it with your supervisor, although he promised to discuss with your supervisor further. But I see that his point is valid, that they can come together instead of separating them into two different, words or terms, while it's still one broad, field of study that you are doing your research. All right, so because when you now go into the study, you cannot begin to have different techniques, different methods, and so on with different terminologies. Okay, so that's my contribution to that conversation. Then the next is your objectives, because if I look at your objectives, I see that it has to do with study, but I don't see, is there a difference between the three of them, because they are still different reproductive organs that I could see. Then maybe when you go back to it, we could see if there are really differences between the first and the third, for instance. Because if they are all the reproductive difference, so it's still one objective there. All right, so please we'll look at that further. Maybe if you can highlight the differences really in this your objectives. Okay, then finally, this interleukin 6 and then interleukin 8. What really is the benefit of this, inflammatory, cytokine? So this, and so the question now is that, I believe you must have studied literature. So from literature and then comparing with the work you have done, is it a lower value of this, markers, inflammatory markers, that is of benefit, or a higher value? So that is the question. I hope you got my question right. Okay, this in the animals at this age that you use for study, is it of benefit to have higher values of this interleukin 6 and interleukin 8, or lower values, compared with literatures and your background knowledge of these markers? Thank you very much.
Thank you very much. Let me start with the interleukin 6. Interleukin 6 is a pro-inflammatory cytokine It's involved in immune regulation, inflammation, hematopoiesis, and tissue repair. It is produced when body responds to infection, injury, or stress. At this juvenile stage, it is expected that the values should be low. When there is disease condition, injury, or stress, there will be higher titration. For interleukin-8 is a pro-inflammatory chemokine involving immune response, inflammation, and set signaling. It is a chemoattractant that attracts immune cells, that is neutrophil, to the site of infection or tissue injury. At this current stage of this animal, they are still at the lower level. Once there's issues, there will be an increase. There will be up-regulation for the titer. Question to you, okay, let me start. What is the benefit of the interleukin-6? Is for diagnostic purpose. If we are able to pin it down at this level, that this biomarker is expressed in this animal, for diagnostic purpose, blood serum level of these biomarkers are useful for the purpose of diagnostic studies.
Okay, maybe you did not understand my question. Sorry. You don't understand my question. My question is this: that from literature, when you compare the value, because are you saying that these values have not been determined in the animals before? Because I wanted to know if the value you obtain in the animals you studied, it's lower than what you came across in literature, or higher, and if that the interleukin-6 and interleukin-8 now, if the value that you obtain in the animal is of benefits to the animals.
Yeah, the value are of benefit to the animal. They are still at the lower level, and basically what we did in this research is immunolocation of these biomarkers. The question is, are they even expressed at this age at all? And according to the report we get from this research, the answer is yes, they are expressed at this current stage of the animal.
Okay.
Can I come in?
You can go. Okay, somebody wants to speak. Yes.
Yeah, can I say something? This brings us to where Dr. Ahu started, the topic. These are actually histological studies. If I'm not mistaken, histochemical studies, they talk more of inflammation. The results are what is our concern, because immunohistochemistry is a technique. It is the results that we are trying to look at. And in this case, you are including inflammatory markers. And so we are looking at inflammation. So the topic with your immunohistochemical studies is really ambiguous. You could just tell us that you're doing some histological studies and inflammatory or immunological profile of the reproductive parts of these juveniles. I think that would have been better than using a very bogus word that happens to be a procedure. That is why you're getting all the questions, because if we're looking at the inflammatory or immunological profile of these organs at this stage, then immunohistochemistry is also very appropriate. That was my initial concern. It's like you're doing 3 different projects in one, and they are not really coordinated, because I became confused at the point of the immunohistochemistry. It is supposed to be telling us a different thing from the histology you are doing. But what it is telling us comes from the histology process that you did. I hope I'm making sense. So that is where this confusion is coming in, and the questions, I expect that you will get more of these questions.
Information.
Yeah.
Information.
Yes.
Information.
Yes.
Thank you very much. You see, regarding the cytokines, not all cytokines are inflammatory. Yes. I would have expected him. There's a very fundamental, crucial, important cytokines. There are a group of cytokines that are known as growth regulatory cytokines. And I personally would have expected him to look at the expression of these growth regulatory cytokines. And a major one of the growth inflammatory cytokines in the reproductive system that is responsible for oviduct epithelial cell regeneration is the interleukin 22. Yes, interleukin 22. That is because cytokines are grouped as pro-inflammatory, anti-inflammatory, and growth regulatory. So for reproductive studies, you don't need the pro-inflammatory, you don't need the anti-inflammatory. The one you need is the growth inflammatory cytokines. If you are to go for any other cytokine, apart from, in addition to these growth inflammatory cytokines, what you should have done is the pro-inflammatory cytokines. And the pro-inflammatory cytokines, the major one in reproduction is interleukin 10. It's interleukin 10. So that is it, because that interleukin 10 not only serves as pro-inflammatory, it also serves as immune tolerance. It tells you how immune tolerant the body, and most especially the environment of the reproductive system is. That is it. So I've not asked my question because this is just to interject so that yes, we continue. Yes, but it just an information that yes, there are cytokines that are not inflammatory, but they are good regulators. Thank you very much.
Oshala, you are yet to answer the question about previous studies by researchers in your department, in that department.
Yeah, there are previous studies, but basically they were in the adult stage. This study narrowed down to the juvenile stage of this grass cutter. There are previous studies that use other immunomarkers, but this study narrowed down to the use of S100, H1C125, interleukin 6 and interleukin 8, Asian blue, and PASS. So this is the first study using these particular markers in this stage of this animal. Other studies were done too, but they are the adult stage of the animal.
That is the more reason why it should be one of the justification of the study. I'm not sure it should be cited in the justification for this study. Previous study was done in adult, and now you have current study is in juvenile. So you need to cite those studies. It's important. Dr. Emmanuel Mbede has a question. Please unmute yourself and ask your question.
He has not answered the question.
Okay, he will do that.
Okay,
His
thank
hand has
you.
been off for a while now. Please go ahead.
He asked a question on the issue of the spelling of uterus. I think that is typographical. The right spelling is UCERUS.
Okay, all right. Dr. Emmanuel Mbede, please. Dr. Mbede. Dr. Mbede.
I agree with what the previous speakers have said, because it is not the amount or the volume of the work. At the end of the day, once you begin to add many things together, you just end up confusing and modeling up everything. You need to have a defined objective. What do you want to achieve? Like the last one, the issue of cytokines. I am still wondering what are you looking for with S100? Is it that you want to map out the neural activities in the reproductive tract? Because I've not really seen, that has not been answered from all your presentation. I expected to see, since you are doing a kind of histomorphological studies, to see the difference, a kind of histological presentation where the different, should I say, the neural cells, neural activities are mapped along the different tissues, but I've not really seen it in this study.
Okay, thank you for your contribution. Dr. Ochala, please respond.
Yeah, basically, the use of S100 is for the purpose of immunolocalization. Now, if you look at my recommendation, the further study should be conducted at different stage of this animal. For us, we have picked the juvenile stage of this animal, and we have successfully determined that S100 is expressed at this age. So it's for the purpose of immunolocalization. Is it even expressed at all in this particular animal and at this age of the animal? So
what do you mean? Sorry, what do you mean by expression of S100?
When the tissues were stained with
expression of S100 is you are trying to target a neural tissues. So the presence of those neural tissue is different from expression. You kept saying expression, expression, expression. So I'm wondering what is being expressed that you are detecting with X100.
I think what he meant is that they are present. They are present at that stage.
They are present.
So and they are not expression.
So it's not really expression.
So Ochala, please take correction. They are not expression, else we should see some data that will show us that they're expression, whether they're protein gene expressions or protein expressions. So please take that correction.
So regarding this, do you have this slide? Sorry, please. The slide you showed, do you have the histological picture? Okay. It's okay. It's okay.
This is for photomicroscope showing diverse expression of epithelial cells, strong expression of neural cells, and very weak expression for stromal cells. And this table summarizes it. The table here. You can see for this particular organ, ovary, you notice that it has the stromal cell and then that of the nerve fibers. Then if you look at this particular table again, at the level of the oviducts, you get that lamina propria, musculari mucosa, and nerve fibers. If you go to that of the uterus, you still get it at level of strong.
It's okay so that other people. I think we are short of time, so that other people. I'm okay with that, so that they can respond.
Professor Sadi's handy. Yes.
Thank you very much. Thank you very much, ma. You see, my brother over there made mention of something that you see. I'm always of the idea that yes, you can do one thing and do it very thorough. So when you say S100, I don't think the candidate really know the value or the function of X100. There are different types. X100 is a protein, and he used a juvenile. And in early pregnancy, the same that S100 protein is being expressed. And what is the implication of that expression? That means that whenever you see S100 protein being expressed, that means it ensures that there is healthy embryonic development, and which also qualifies successful implantation of the embryo. However, there are also other variants of S100, like the S100A4, S100A2. This S100A2, their expression is not a good one. Does not show that yes, and that's what the prof is saying. For any cytokines you are measuring, tell us when the expression is there, what does it imply? When the expression is not fully expressed, what does it imply? So I could have of the view that yes, that expression of this. Like, for instance, when you are looking at progesterone, progesterone signaling, and that's why you look for interleukin 22. For any interleukin, whatever you are going to assay in any research, you must have a reason why you are doing that. Not just, a cascade of several interleukins that, yes. Just, in fact, if I were you, I can measure one cytokines and I will do so many things under it, and that will give me my degree. So not just taking different things, and that does not even tally to what you are looking for. So instead, yes, tell us the implication of each of the cytokine and what have you. So the variant, when you say which, you might not even be which antibody did you use for the immunohistochemistry? Just answer the question. And to my colleagues over there, the title histology and immunohistochemical can stand. Please, yes, and it has been a standard something. Yes, it can stand. I'm not, at least, I'm not his supervisor, but yes, several things, and these are two entirely different areas. Immunohistochemistry is a technique. It's a study on its own. Thank you very much.
So, Dr. Charlotte, answer the question. Which antibody?
The antibody used is mice monoclonal antibody.
What do you say?
Mouse monoclonal antibody.
Haba. Monoclonal or polyclonal? Haba. Monoclonal or polyclonal?
Poly rather, sorry. That was that was a slip of tongue. Polyclonal.
If you have any good grasp of what you have done, immunohistochemistry, in fact, let me ask you another question. Where did you do the immunohistochemistry?
I will chat you up, sir, for the details.
Where did you conduct it? Where did you do it?
Where exactly?
Hello, sir.
Answer the question here, Peter Ochola.
The preliminary sample collections were done at the histology lab of the Department of Veterinary Anatomy in Justin. Then the secondary phase of the work was done at National Hospital, Department of Histopathology, National Hospital in Abuja.
No, the immunohistochemistry?
Yes, sir.
MD Sani, can I make some contributions now?
Yes, sir.
Yes, just in respect to the immunohistochemistry. Well, in my layman understanding, it's a study, but I think it's also a staining technique using antibody-antigen reactions to produce to generate some color that are being stained. So when you combine histology and then also immunohistochemistry, why did you just stand? Why just one of these titles?
One house. Who is speaking, please? One person.
And
are we still?
Yeah, Dr. Victor, hello? Yes. Yeah, we can hear you.
Yeah, we can hear you.
Hello.
We can hear you. Go ahead. Dr. Victor, are you there? Are we together?
Okay, so I was making. Yes, I am. Am I still audible? Yeah.
Yes, we can hear you. Go ahead.
Okay, so thank you so much.
I think it's
my contribution was to what
the network is playing up.
Hello.
Yes, your network is playing up. So can you kindly meet the supervisor and the student after this to discuss something?
Okay,
Thank you.
I'll do that.
Thank you. All right. So I think the questions have been exhausted.
Okay.
Yeah.
I have my own comment.
Right.
There was no conclusion in the presentation. He just jumped into.
Okay.
There was no conclusion. So what was the conclusion from his work? And Dr. Chada, what can you actually tell us from your work to in a layman's language, a non-technical language, what is your conclusion from your work? What is it that you can tell us that you have achieved in your work? Please answer. Dr. Challa, are you here?
On the study, we were able to demonstrate the normal histoarchitecture of the reproductive organs of the African greater gerat at this particular stage. We were able to demonstrate the expression of acidic mucopolysaccharide using Asian blue. We were able to demonstrate the expression of neutral mucopolysaccharide using periodic acid sheet. I was able to demonstrate the expression of interleukin 6, interleukin 8, and calcium to the 1 to 5 and S100 at different epithelial cells in this particular stage of the animal.
Okay. So conclusion from your work and other previous work, what can you conclude from your work, from your research? What do you conclude based on other previous studies?
Yes, basically on the histochemistry and the normal histological part of the study, I agree with other researchers. But so far, there's no published data on interleukin 6, interleukin 8, calcium 1 to 5, and S100 in this stage, juvenile, of this particular animal species. So I would say this is the first and novel information revealed by this research on the expression of interleukin 6, interleukin 8, calcium 1 to 5, and S100 in this species, the African greater gerat at the juvenile stage. Other research can be conducted to the neonatal stage, the adult stage, and then there could be a cross-sectional study to bring all the stages, the expression at different stages together. But so far as of today, no work have been published on this particular markers.
Professor Bosha's hand is up. You can, you have the floor.
I want to make these comments. One, when the Professor Sani asked a question and the presenter said you will chat him up properly, that is not the right thing to do. When somebody asks you where you do your research, under this condition, you are obliged to answer the person, not to say we chat you up privately. This is not a secret thing. It's not as if you do something in secret and you want to hide. Nobody is saying you should release your patent to him. So don't ever answer somebody like that when they are asked, where do you carry out your research? It's not right. You should be able to tell the person.
Thank you, sir.
Chairperson, you have.
Secondly, we have somebody is sharing it, and there is a seminar coordinator. So if you are communicating, it shouldn't be a communication between you and the other person making comment. You direct your question to the person sharing, so that he or she will know how to pass the information. Let it not be an intercommunication between the person asking question and the other person trying to respond. We should be able to keep to the etiquette of some of these things. And finally, my other comment is that concerning the topic, as others have said, I don't want to agree with Professor Sani made a comment that histology and immunochemistry can stay. I agree with that. But the fundamental
Are we together? I can't hear you.
Are you hearing me?
Okay, I hear you now.
Okay, I'm saying the topic is saying histomorphology and then immunochemistry. Professor Sani said histology and immunochemistry can stand. But I don't want to agree that histomorphology and immunochemistry can stand, because the work is strictly histological work. The way I understand morphology, if you are talking about morphology or morphological thing, you are talking about the growth structure of those things. So say histomorphological and immunochemistry is a misplaced statement. That is not right. That topic has to be reframed.
Okay.
Except people are convincing me that morphology and histology is the same thing. That work is purely a histological work. There is no aspect of morphology. We talk of morphological this thing. You are looking at the growth structures and some of these things. I think there is nothing gross on those things he has done.
There are many histologies.
Mainly histology. So what is histomorphological about it? So that topic has to be tempered with. There is nothing morphological there. So the morphology has not to be there. And we cannot overemphasize it. There are baseline information that some people, even in the college, in the department, has established. Reference should be made to such things and all these things. That's just my closing.
Thank you very much. Am I being, sir?
Yeah.
Actually, you are absolutely correct. Instead of that histomorphological, if the candidate and the supervisory committee feel that the histology should be there, it can just be shortened as histological. instead of histomorphological, yeah, it will just be histological.
Thank you. Thank you.
Yes, sir. Thank you very much, sir.
It's all right.
And again, you see, the candidate, just one question, last one. You see, that's your conclusion. I'm not convenient with the conclusion because you cannot tell that whether the level now you obtain in the expression of those cytokines and whatever that you analyze in the juvenile are normal. You cannot tell until just one single experiment. You cannot say, okay, yes, okay, this is what you concluded, because this level, more especially those cytokines and that S10. In fact, how I wish you know the value of S10, in cancer, in cancer progression. I'm telling you, in fact, even in cancer progression, S10 protein is one of the proteins that is usually looked for when you are looking for how tumor progresses and what have you. So that expression now, is it normal? Is it abnormal? Is it overexpressed? You cannot tell at this level. Thank you very much.
Thank you. I want to appreciate all of you for your contributions, your questions, and I would like to invite the supervisors to make a brief comment. We have seriously run out of time. So over to you, supervisors.
Dr. Rachel.
I saw Dr. Rachel. She was present.
Yeah.
Please, over to her. She can make her comments so that we close for the day.
Please unmute yourself. I've asked you to unmute. Dr. Rachel Kozeze, please unmute yourself. You can unmute yourself now.
Hello, good afternoon.
Good afternoon.
Can you hear me?
Yes.
Yes, we can.
Hello?
Go ahead, we can hear you.
Okay. Good afternoon, the PG, the Dean of the College, other members, professors who attended this today. On behalf of the other member of the team, I would like to appreciate everyone at home. I want to appreciate to this work. I must say that continuous and knowledge is also continuous. So taking these contributions for granted, but I'll just highlight a few of some of the key contributions that emerged about. For instance, the topic, the supervisor go back and look at it. No, but I just want to say that histology is different. So in this study, we felt like being specific about the kind of histology we're trying to do. So on that topic, we will go back and though we see a make the topic very specific. And for some of those presentations, agree at the supervisor that the candidates, a lot of things, and some of those things were going to be affected before they walk the package presentation examination. So once more, thank you for your very valid contributions to this work. I'm very sure that it has improved the quality of the work. The candidate is going to sit down together with you most of this. Thank you.
Okay. Professor Samuel there, does he have anything to say? If no, I want to thank you for your time, your patience, your contributions. We look forward to exciting presentations like this. And to the supervisors, I'll say, always be straightforward in your research and do what you intend to do. To still give a student their postgraduate, you don't have to bring in too many parameters. Thank you so much. I now hand you back to the coordinator. Thank you.
Thank you very much, Ma. I want to use this opportunity to thank each and every one of us that have made our time to be here today. We started around 10:10 and we are still here 12:48. Thank you for your contributions. Thank you, Professor M.D. Sani. Thank you for coming in. We appreciate having you. And we hope that next time you will make it a date with us. I also want to thank those of us that are not within Makurdi that have made themselves available. Dr. Emmanuel Mbede, thank you for showing up. Dr. Ben Onoja, thank you. And some of our students who are on transit, Dr. Sese, thank you for showing up. I also want to thank the other postgraduate students, professors present, senior colleagues. Thank you for showing up. The next seminar, I will communicate to us the next seminar. So presenters, please make contact with the chairpersons to collect the hard copy. They have made corrections on the hard copy. So make effort to collect back the hard copies from them so that you can effect those corrections on your thesis. At this juncture, I also want to thank the dean, thank other members of my committee, Dr. Joseph Abo, Dr. Peace Kumate, and others who made themselves available for this seminar. We will continue in this fashion for a period of time until we're able, until we are sure that the road to campus is safe for us. So you will plead with us that we'll continue this way virtually until we're able to get ourselves into the campus safely. Thank you once again and have a wonderful day. Thank you. We have come to the end of the seminar. So do have a wonderful day. Thank you.
Thank you, everyone.
Thank you, sir. Thank you for coming.
Bye-bye.
Thank you so much. We appreciate the session.
Thank you. Thank you very much.
Can we now?
Yes.
Can we go up now?
Yes,
Yes,
you
Mom.
can exit now.
You can exit now, yes. Okay, thank you so much. Mm.